Hello, and welcome to our All of Us Science Day. This is Poster Lightning Talk 3B, Understanding Specific Health Conditions. My name is Nicole McNeil Ford. I'm a program officer in the All of Us program at NIH. I work in the Enrollment Coordination Center and Implementation branch, and I will be your moderator for this session. Before we get started, I wanted to go over some housekeeping information. So we're here to help. If you have a need for closed captioning can be turned on by pressing the CC icon in the top right corner of your video. If you run into any technical issues, or need any accommodations, please email Science Day, All of Us Science Day at pyxispartners.com. And we want you to participate in the conversation. To ask a question, select the Q&A option at the top right hand of your screen and type in your question and then submit it. Questions submitted through the chat box will not be monitored. Please use the Q&A box if you have a question. And for this session, all presenters will focus on understanding specific health conditions. So I'm delighted to introduce our panelists today. Join me in welcoming Jonathan Liu. Jonathan has a BA. He's a medical student at Brown University. We have Bree Groves, is an undergraduate student at the University of North Carolina, Chapel Hill. We also have Hannah Siegel, bachelor's, is a pre-doctoral graduate student at Vanderbilt University. We also have with us Isaac Wade, is a data analyst at the University of Oxford. We also have Ijeoma Nnadozie. She's a pre-doctoral graduate student at Keck Graduate Institute. And we also have Uri Bright, a postdoctoral research fellow, postdoctoral at Yale School of Medicine. So during this session, we will have our first three speakers. Those three speakers will present, and then we will have Q&A. And then after that first Q&A session, we will have the final three presenters. We will then wrap up with Q&A for the final three presenters. If you have any questions, again, put your questions in the Q&A section, in the Q&A box, and we'll get to them during that part of the session. So right now, we will begin with our first presenter, Jonathan Liu.
Hi, everyone, and thank you, Nicole, for that introduction. Today, I'll be presenting a work on the diagnostic timeline from ocular prodromes to myasthenia gravis. First, for some context, myasthenia gravis is a chronic disorder where one's own antibodies attack muscle receptors. This results in fewer receptors that can receive neural input, and as you can imagine, if our body is receiving fewer neural inputs from fewer muscle receptors, we begin to experience muscle weakness, and because of that, a characteristic hallmark of myasthenia gravis is a fatigable muscle weakness. This means that the muscles worsen with use, and because they are constantly working and have fewer receptors than other muscles in our body, the eyelids are often the first to display this weakness. However, there are many causes for weakness in muscles around the eyes, which can make diagnosing myasthenia gravis difficult in clinical practice. And because I mentioned that myasthenia gravis is characterized by a fatigable muscle weakness, clinical studies show that early treatment can preserve more of that neuromuscular function and also reduce disability. Since isolated ocular symptoms can be really hard to pinpoint to a specific neuromuscular condition, our study aimed to better understand how often these ocular symptoms progress to myasthenia gravis and also how long that diagnosis takes. To accomplish this, we constructed two cohorts. one cohort with 11,966 patients with ocular symptoms of ptosis, which is known as eyelid drooping, diplopia, also known as double vision, and strabismus, which is a misalignment of the eyes. Our second cohort was of 433 patients with diagnosed myasthenia gravis, comprising ocular, generalized, and also adult forms. The first finding that I'll direct you to is that across the cohort of patients with myasthenia gravis, more than 97% of them experience at least one of the ocular disorders I previously mentioned. However, across just that cohort of patients with ocular disorders, just less than 2% of them resulted in a myasthenia gravis diagnosis, once again showing that ocular disorders are highly prevalent in myasthenia gravis, but on the flip side, ocular disorders are quite generalized and a little bit hard to pinpoint to specifically myasthenia. Now, these are general findings without any temporality, and as I previously mentioned, we wanted to examine the timing of progression from ocular disorders to myasthenia gravis. So from our initial two cohorts, we isolated a subgroup of 221 patients who had an ocular disorder diagnosed before the first instance of myasthenia gravis in their health record. If I'll request moving to 2 slides forward, please. Here is really what we found. In the top left panel, we looked at the number of years it took from the initial ocular presentation to myasthenia gravis diagnosis. And in that top right, you see the same figure shown within just that first year. Highlighted, you'll see that the median time to diagnosis of this cohort overall was 121 days, or approximately 4 months. And if you look at the bottom two panels, we did an extra little insight where we wanted to see whether there was a difference between diagnostic pathways between males and females assigned at birth. And so we found that female patients on average received a diagnosis of myasthenia gravis about 3.5 months later than male patients. And this was, I think, the most striking outcome that we really had, where we found that there was a much shorter latency of diagnosis among male patients, and really wanted to see, does that mean that the diagnostic pathway is really shorter in male patients, or do we have a diagnostic delay in female patients? And so we wanted to see this in a few different ways. The first way that we did, if you go to the next slide, is a distribution visualized where we looked at the ages at which men and women first developed ocular symptoms before they went on to get myasthenia gravis. We did this clinically because looking into the literature, there is actually an earlier peak of onset of myasthenia gravis among women and a later peak onset among men. This is a bimodal peak of distribution that has been quite replicated in the literature, and over in our project as well, we see the similar thing. With men on average presenting with a diagnosis of an ocular symptom around age 63, and women presenting around age 54. And so just for this final slide, I'll show you kind of with all this information that we found, kind of what those clinical implications are. First off, we see that a significant majority of myasthenia gravis patients do experience ocular disorders, but at the population level, really myasthenia gravis following an isolated ocular symptom is not super common. Among people who do develop myasthenia gravis from an ocular disorder, that timeline is about 4 months, and we do see that male sex is linked to a significantly shorter transition. While we weren't able to really get to the exact reason for why this difference is occurring, we do believe that there is an association with female patients just by demographic profile being younger, having a lower clinical index of suspicion for myasthenia gravis. So we do urge clinicians to remain cognizant of this demographic profile to make sure that we don't overlook ocular symptoms, but at the same time also being cognizant that it's not very common at the population level. So thank you so much for your time, and I'm happy to answer any questions.
Thank you, Jonathan. Next up, we'll have Bree Groves.
Hi, everyone. My name is Gabrielle, or Bree Groves. Today, I'll be presenting research on eating disorder diagnoses and treatment patterns using the All of Us dataset. You can observe the research poster characterizing self-reported eating disorders in the All of Us research program shared on screen, and I will walk through study objectives, research process, and findings. You can continue to the next slide. So eating disorders such as anorexia nervosa, bulimia nervosa, and binge eating disorder are serious mental health issues characterized by disruptions in normative eating patterns and often an intense fear of weight gain or changes in body shape. Eating disorders affect individuals across the lifespan, but what we are better trying to understand is who gets diagnosed, when they get diagnosed, and whether they receive treatment. Most of what we know comes from clinical samples or nationally representative surveys. While valuable, those approaches don't always capture the full diversity of real-world lived experiences. Our goal was to explore the All of Us research program datasets as a different kind of resource. While it is not nationally representative, it provides a large and diverse sample that allows us to examine patterns of diagnosis, demographics, and treatment experiences. This matters because improving our understanding of who is diagnosed and when can help identify gaps in care and opportunities for earlier, more accessible, and more effective intervention. Next slide, please. Our main objective was to use the All of Us dataset to examine differences in demographic characteristics between individuals who reported an eating disorder diagnosis and those who did not, as well as to explore diagnosis timing and treatment patterns in those endorsing an eating disorder diagnosis. We conducted a descriptive analysis looking at variables such as sex at birth, educational attainment, age at diagnosis, and current treatment use. One strength of this dataset is that there is relatively strong completion across variables, which allows for meaningful comparisons. However, there are important limitations to consider given that the data is self-reported. We cannot distinguish between different types of eating disorders. Additionally, while the dataset is large and diverse, it is not nationally representative and is skewed towards certain groups, particularly females and white participants. All this considered, our findings should be interpreted as more exploratory rather than definitive. Next slide, please. All right, so I'm going to take a moment to walk through what you're seeing on this slide. First, directing your attention to Table 1, this summarizes the demographic differences between individuals with and without reported eating disorder diagnosis. Overall, we found several patterns consistent with existing literature. For example, there is a significantly higher proportion of females among those with eating disorder diagnosis. An important consideration is since the data itself is skewed female, this may partially reflect the sample rather than the true population differences. We also observe a small yet interesting difference in educational attainment, with about a 5% lower rate of less than college education among those with eating disorders. This could relate to typical age of onset, which overlaps with the transition to college, though more research is needed to better understand this relationship. If you take a look at the figure on the right, this is where things become more complex. To orient you, this shows current treatment use by age group at the time of diagnosis. The most immediate insight was that the largest proportion of individuals falls into the neither category, meaning that they are not currently receiving treatment. Despite being diagnosed at a time when symptoms are emerging, the adolescent group for age of diagnosis shows relatively low current treatment utilization. This raises an important question about treatment use. Does diagnosis earlier in life combined with lower current treatment reflect appropriate non-use of treatment or unmet needs? On one hand, some individuals may have received treatment earlier and no longer require it. Those who are recovered or in a state of recovery not requiring treatment. Alternatively, it is possible that a substantial portion of individuals in the eating disorder group may need treatment but are not receiving it. At this stage, we cannot distinguish between these possibilities, and the pattern suggests that treatment might not always align with clinical needs. This reveals an important area for future research. We also observe treatment needs tend to increase somewhat into adulthood, which could reflect ongoing care needs or re-engagement with treatment later in life. This figure highlights the overall complexity of understanding treatment utilization across the lifespan. Next slide, please. Overall, our findings highlight both the value and limitations of using the All of Us dataset to better understand eating disorders at a population level. One key strength of the All of Us dataset is the ability to link survey data with electronic health record or EHR data. This creates an opportunity to better understand how self-reported diagnoses align with clinical data and develop a more complete picture of individuals' health experiences. An important next step in this work would be to leverage the EHR data to better understand the underlying clinical picture. This helps us distinguish between individuals who are currently receiving treatment because they no longer need it versus those who might still need care but are not receiving it. Importantly, our results point towards potential gaps in treatment, especially for those diagnosed later in life, which could have long-term implications. If individuals are diagnosed but not treated, it may contribute to longer duration of the condition. These findings also connect to broader goals of improving equity and access to care by identifying who might not be receiving a diagnosis or treatment, and we can better target interventions and improve outcomes. Ultimately, this work supports the idea that large, diverse datasets like All of Us can complement traditional research approaches and help generate new hypotheses for future studies. Thank you.
Wonderful. Thank you so much for that, Bree. Next, we will have Hannah Siegel.
Hello, everyone. Today, I'll be presenting my research on blood pressure genetics in over 2.5 million people. First, I'll start with, next slide. Thank you. First, I'll start with just a quick overview of kind of what genetics is. So our bodies are built and run using instructions stored in DNA. And we get most of our DNA from our parents, which is why health traits can sometimes run in families. These DNA instructions help control things like how our bodies grow, how organs work, and how our cells respond to stress or disease. And even though we get the majority of our DNA from our families, each person's is a little different. Those small differences can help explain why some people are more likely to develop certain health problems than others. In this project, I studied how those DNA differences are related to high blood pressure. Next slide, please. Blood pressure is the force of blood pushing against the walls of your blood vessels. When it stays too high for too long, it's called hypertension, and nearly 1 in 2 individuals in the United States has hypertension, and it increases the risk of serious health problems like stroke, heart disease, and kidney disease. But hypertension doesn't affect everyone equally. Some groups of people have higher rates of hypertension and more blood pressure-related complications like stroke and even stroke-related death. These differences are shaped by many factors, including daily life, access to care, environment, and biology. And so one important question is, can genetics help us better understand why blood pressure differs from person to person and across population groups? Next slide, please. To answer that question, we used data from more than 2.5 million people across 16 different studies. And one of those was the All of Us research program, which could substantial amount of data from non-European populations. And that's especially important because most genetic studies have been historically focused on people of European ancestry. I studied 3 blood pressure traits: systolic blood pressure, or the top number, diastolic blood pressure, or the bottom number, and pulse pressure. Each of those traits gives slightly different information about blood pressure biology. Then I looked across the genome, which is another term for all of our DNA, to identify DNA changes that might be linked to blood pressure. I studied everyone together and also looked at different population groups separately. That allowed me to ask 2 main questions. First, which changes in DNA are linked to blood pressure? And second, are some of these signals shared across groups, while others appear more strongly in only one specific group or specific groups? I also included the X chromosome, which many earlier blood pressure studies have not included, and we also received a lot of X chromosome data from the All of Us research program as well. Next slide, please. Here are some of our main results. So on the left, this is what we call a Manhattan plot. Each dot represents a DNA change, and taller dots show stronger evidence that this change is linked to blood pressure. The big takeaway is that blood pressure is not controlled by just one gene or one small area in the genome. It's influenced by many parts of DNA all across our genome. And so in this study, we found thousands of blood pressure linked regions, including many that have not been reported before. And on the top right, this table shows that we identified important genes or regions of the genome in every population group that we studied, and some of these genes were unique to specific populations. On the bottom right is a close-up of one DNA region located on chromosome 20. In this example, the labeled signal, a specific change in DNA, only showed up in the Asian analysis, which is represented in blue. And so that highlights one of the main findings of this project, that some parts of blood pressure genetics are shared across groups, while others are stronger in specific populations. Next slide, please. This work matters because high blood pressure is still a major cause of preventable disease, and many people still don't have their blood pressure under control. That can lead to serious health outcomes like stroke, heart disease, kidney disease, and death. By studying genetics in a very large group of people from different populations, we can better understand the biology behind blood pressure. In the future, that could help researchers build tools that help predict who might be at higher risk of developing hypertension, identify new drug targets, and support better treatment. These efforts could lead to prevention of uncontrolled blood pressure for millions of people. And this study also shows why it's important to include many different groups of people in research, especially when the studies focus on a health outcome that disproportionately affects some populations over others. If research only includes one group, we may miss important parts of the picture that could be instrumental in development for better treatment options. And so overall, this project helps build a stronger and more complete understanding of blood pressure genetics, thanks in large part to programs like All of Us, where participants generously contribute their genetic data to help researchers better understand disease and hopefully improve health for millions of people. Thank you.
Terrific. Thank you, Hannah. So now we can begin our first Q&A portion of the session. Once again, if you have any questions, please enter those in the Q&A section, in the Q&A box. All right, so we have a question. First, I want to thank the presenters. They were excellent presentations. For Hannah, one of the questions came in for you. Did you control, or was it possible to control for genomic inflation, adjusting with lambda in the multi-ancestry loss?
Yes, great question. So we didn't adjust with a lambda, but we did use principal components, which were generated within population groups. And we did also look at, we performed LDSC and looked at the lambda, and there was no inflation that we expected to be due to any sort of population stratification.
Thank you. There's a question that came in for Hannah again. How did you evaluate if a locus was ancestry specific and novel, considering that locus definition is variable across many different studies? And then there's a second part. Well, maybe I'll let you handle that first part.
Okay, yeah, sounds good. So that's kind of a two-part question. So for ancestry specific, we performed fine mapping using SUSY-X. And then the slide that I showed where it kind of had a list of number of genes that were specific to ancestry groups, that was done using, perfect, thank you. Those genes are listed from genetically predicted gene expression, which we generated using SPredictScan, and those numbers are just whether or not those genes were significant in any other population group. And then, but we also did fine mapping for lead loci. And then for considering novel, we identified our significant independent loci using clumping with Plink. And so we considered a SNP novel if they're all known published loci, if there were any in a clump with one of our lead loci, we didn't consider that as novel. So to be novel, it could not be in a clump with another known locus. If that answers that question.
Yeah. Super. I think I will direct a question to Jonathan. Jonathan, you showed that MG had a trend of the sex difference to males. Did you find any other factors, such as like environmental factors or behavioral factors, that might play a role in MG?
Yeah, thank you for that question. We were specifically looking just at a general, more population level, and actually didn't look into specific other predictors in that kind of sense. So that could definitely be something that we look at further. We were just more looking at more of a clinical kind of picture, and that's why we focused on demographics and then also outcomes related to just diagnosis. But thank you for that question.
Sure. I have one question for Bree. What motivated you to conduct your research?
So eating disorders are often underdiagnosed and undertreated, despite having serious mental and physical health consequences. And much of the existing research comes from clinical samples, which don't always reflect the full population. So I was motivated to explore whether a large, diverse data set like all of us could offer new insights into who is being diagnosed and whether they're receiving care. The goal is ultimately to contribute to a better understanding of potential gaps in diagnosis and treatment, and ultimately support more equitable access to care.
Fantastic. Well, thank you for that. Thank you all for your presentations. We'll now move on to the next set of poster presentations. And to start, we have Isaac Wade.
Hello, good afternoon or good morning, depending on which time zone you're in. So today I'm going to be going over some of my research using the All of Us platform, looking at the genetics of idiopathic hypergonadotropic hypergonadism. So if you could go on to the next slide, please. So IHH, quite a sort of scary term, but essentially all it is a rare form of infertility. It affects affects approximately 1 in 10,000 men and 1 in 50,000 women. It's relatively straightforward to treat. However, sort of a lot of patients with IHH don't have a genetic diagnosis. So sort of with the current knowledge that there is, about 30% of patients can sort of have an identified gene change that suggests sort of why they have IHH. So the aims of our research project can sort of be thought of as two sides of the same coin, if you can put up with this tortured visual metaphor. So on the one hand, can we identify this gap in the knowledge of genes involved in IHH? And also specifically, can we identify any sort of common variants that are found in lots of people, and whether they have any contribution to such a rare disorder? And then on the other side of the coin, so answering this question of whether there are any of sort of shared mechanisms of IHH and infertility in general with people. So if you could go on to the next slide, please. So what we did, so Hannah's sort of very well explained sort of what a GWAS is and what the results of that look like. So because we have so many cohorts at a sort of essentially a national scale, we have a significant sort of case series to look at the genetics of a disease as rare as IHH. So yes, we have a meta-analysis, a meta-GWAS of 7 different cohorts, and that's what this map is showing here, including all of us, but we also have sort of a couple of cohorts in the UK, one of which is a South Asian cohort. We've got Estonia, we've got Denmark, Finland, Iceland, so sort of, yeah, biobanks databases all over the world. So we look at all of the variants and whether there's any sort of genetic changes that are identified more often in people with IHH than people without IHH. So in total, we had 3,000 cases, and then comparing that to 1.5 million controls. So if you could go to the next slide, please. So the main finding, so we find 4 sort of positions in the genome where variants are associated with IHH. 2 of these sort of independent variants implicate a new gene, so that's shown at the top of this slide here, C1q TNF8. So, and yeah, we also sort of followed that up and found gene expression data that sort of adds to that evidence, and that's sort of gene expression. You can see, so yes, it's like how different areas of the body sort of express genes at different levels. So sort of areas that we're interested in, the gonads, and then also the brain, express this gene quite highly, which suggests that it does have a role in IHH. And then this other, or one of the other hits that we find is in or near SHBG, which is a hormone involved in the regulation of testosterone and estradiol. And then when we actually looked at this variant in the general population, so not just in people with IHH, we find that it does have a significant effect on how much sort of testosterone you have at any given moment. So next and final slide, please. So hopefully I can convince you why this matters. So yeah, our main finding, we find 4 common loci that are associated with this condition, IHH. It's a relatively modest addition to the gap in the knowledge of the genetics of IHH, but hopefully helpful for patients if it's confirmed in an independent data set, and then helpful for their families as well. And then hopefully I can convince you that there's some wider implications to these results. So specifically, this variant in SHBG having effects on estradiol and testosterone, that could help to deliver on efforts for personalized medicine. And then even more generally than that, sort of these methods, these GWAS methods are quite well established. They're used in lots of different ways. As population cohorts, like all of us, continue to recruit more and more people and get better linkage to healthcare records, then these sort of methods that have been around for 20 plus years can be applied to rarer and rarer traits, which is quite an exciting thing. So thank you very much for listening.
Great, thank you, Isaac. Next up, we have Ijeoma Nadozije.
Hi, everyone. Good morning. My name is Ijeoma, and today I'm going to be talking about a critical, often overlooked colorectal cancer research disparities, which is one of the real consequences of millions of Americans, especially African American. As I earlier said, my name is Ijeoma, and this is a story of genetics, equality, and what might we be missing when we only look at one group of data. Our study is a comprehensive cross-ancestry genomic framework for colorectal cancer, integrating common coding and rare variants analysis using the All of Us program. Next slide, please. Next slide. So let's start with the problem. In America, colorectal cancer disproportionately affects African Americans. They have a higher incident rate, higher mortality rate, and are often diagnosed at an advanced stages compared to the European Americans. Yet, despite bearing these heavy burdens, Africans are disproportionately underrepresented in genomic studies, and that's why I wanted to highlight this group. More than 80% of genetics GWAS studies that are done are conducted in solely European populations. This means that the risk models, screening tools, as well as genetic markers that are used today are predominantly excluding this population. There are 3 critical consequences here because we have the current risk factors that are primarily built on European ancestry GWAS data, ultra-rare variances that are more common in Africans and are by the conventional studies that are currently being done, and ancestry-specific variances that are dangerously underrepresented. This is not just a scientific gap, it's also a genetic equality problem. So currently on a preliminary level, but so far, what did we do and how did we address this? First of all, we leveraged the whole genome sequence data that was obtained from 250 participants from the All of Us data program. And this was curated by the All of Us programmers and into what we call the All of Us database, which is a Hail-Matrix table. We only selected 2 populations. First of all, we selected 133 European ancestry population, as well as 66,000 African ancestry population. We implemented 4 analytic work frame. First, downloading this into our Python platform. It initially comes as an Hail-Matrix table, and then we went ahead to functionally these variances, classify them into structural variances or indels, and then stratify them by their minor allelic frequencies, predictive functions as well. Then we evaluated the chromosome level variant density, the allelic frequency count, the frequency stratifications, as well as the distribution of variants across the different effects in both populations. We finally tested on multiple different tests, such as the Bonferroni correction, as well as the Benjamin Hux false discovery rates. Finally, we assessed the variant's pathogenicity using the Revu score. Our objective was to identify either specific or shared colorectal cancer variances, compare their variance consequences across these two populations, and then uncover both novel African genes that are exclusive to this population. Yeah, we found in that table, you can see that we found, highlighting the results from the table, we found that there are several genes that show that, which literature has already shown that are predominantly found in the mismatch repair and Leigh syndromes that are highly identified in the European populations, and they were predominantly found in the pathogenic loss of function variants. This confirmed that our approach is working, but however, the African ancestry group, the story was quite different. We identified several genes that do not appear in the current GUS studies or even risk models that are being designed. Only one gene, which is the MLH1, was significantly shared by both populations, confirming that there are cross-ancestry shared groups in our signal so far. And like I said, notably, this is just a preliminary study. This is our findings so far. We also observed notably higher ultrasound burden variants using the minor allele frequencies for higher in African Americans compared to the European Americans, which is a spectrum of variants that often entirely missed by the conventional GWAS approach. Now, the two genes that we found that were strikingly highlighted in the African American population is the LIPDIP8, which is a Ly6 power domain 8. This is a tumor suppressing gene that helps in protecting the gut lining and keeping the bacterial lining well distributed. And its level is much lower when you find it in stage 2 or stage 3 patients with colorectal cancer. However, it's shown that increased level in colorectal cancer cell lines shows a tumor suppressing function as well as enabling cytokine inflammatory signals to be observed in it. Secondly, we found a carotene 20, which is a marker for normal cell development and progression, commonly found in colorectal cancer cells. A lower cytokine 20 levels is associated with a microsatellite instability tumor. It's important to note that synonyms and missense variants were elevated in the REVU score across the African cohort, and specifically gene suggests that real consequences have not been experimentally evaluated. The result of these findings just highlights that we can see that there are common shared genes that are in both populations. However, the African Americans have shown that there are several genes that have not been identified in the current GUSs that have been done across. And this shows that we need to go ahead and do functional and experimental studies on these genes. Maybe this could highlight why we find this burden significantly in this population. There are future studies for this direction. We're still going to implement further pathogenic scores such as CAD and SPLICE AI, as well as expand our studies on other populations such as the Hispanics and Asian Americans. And finally, we would like to implement or revalidate or recalibrate the current polygenic risk scores that are currently being used for colorectal cancer. At the end of the day, colorectal cancer is not just a one-size-fits-all. Thank you.
Thank you, Ijoma. Next up, we have Yuri Bright.
Good.
All right. Hello, everyone. So today I'll present our study about the genetics of erectile dysfunction, sexual dysfunction in men. A study that we conducted using all of us data and meta-analyzed it with previous publications about erectile dysfunction. I'll say a few words about erectile dysfunction and what it is. We can go on to the next slide. Thank you. Very generally, erectile dysfunction is associated mainly with arterial insufficiency, but as you can assume, this is also associated with many other factors, psychological, hormonal, physiological factors, and all of these factors eventually have their interaction with genetics. And common treatment, namely Viagra, are pretty efficient, but only for about 60 to 70% of the population. Therefore, this not life-threatening issue, yet well-being hampering issue of erectile dysfunction can use new studies, new investigations, and new treatments. And the way we did it, we can go to the next slide. Very basically, already presented by previous speakers today, genome-wide association study. Just very briefly, GWAS is a method that we use to scan the entire genome to look for genetic variants that are associated with a specific trait. We compare the DNA of people who have a specific trait and people who do not have a specific trait. If you can press. Thank you. For example, a GWAS of erectile dysfunction will compare the DNA of people who have erectile dysfunction and people who do not have erectile dysfunction, looking for those genetic variants that are statistically significantly associated with this particular trait. We can go on. That is, for example, here we did a GWAS and a few post-GWAS analysis. I will not have time to present most of them here. Some of them are in the poster, other in the publication. But I will present in the next slide our main results. So this is a Manhattan plot. Let's remove that one. Thank you. A Manhattan plot, as was said before, the x-axis represents chromosome and base pair. The higher point is on the y-axis, the more significant the result is. In total, all of the annotated genes here are genes that we found as statistically associated with erectile dysfunction. We have a total of 40 different genomic loci, which proves again the point that erectile dysfunction is a polygenic trait. It is associated with multiple factors. Some of these genes have hormonal roles, others are associated with psychological traits. What you see above all of them, up there on chromosome 6, is a very interesting region, and it is interesting because we really don't know much about it. This is a non-coding region in the DNA. It is known as a regulator of a gene called SIM1. The role of the protein that is coded by this gene is not really known, but it was found as associated with obesity. It was actually found as associated with sexual function in animal studies. And very importantly, we found this effect in all of us, and it was also found in 3 other publications. And it was when we meta-analyze all these results, we got this Manhattan plot right here. So the effect is definitely real, and it's really interesting yet to understand why and how exactly this genomic region affects erectile dysfunction. If we can move forward to the next plot, this is the genetic correlation analysis that we conducted, an analysis that aims to compare or to evaluate how much the genetic architecture of one trait is similar to others. Everything you see in green here is significant, and also here you can see there are a variety of effects, a variety of associations between erectile dysfunction and substance use traits, with psychiatric traits, for example, depression, suicidality, PTSD. There are also a variety of associations with general health traits like blood pressure and pain. Another kind of proof that erectile dysfunction is indeed a polygenic trait and is not merely a trait that is derived only from a specific physiological factor. So I'll use the next slide basically to sum up the main results. As I said, not a life-threatening matter, but definitely impairs life quality of many men worldwide and their partners. Erectile dysfunction becomes more common as men get older. And even though Viagra and other Viagra-like medicines are effective, they are not effective for everyone. And our findings really emphasize the need to investigate deeper the SIM1 gene and the protein encoded by it. And these loci that regulate its expression and how they are associated with this trait. and maybe that can be a really interesting and new target for potential treatment one day for erectile dysfunction. I will say that all the results I presented here are, as other people said, from European subjects, GWAS. A lot of GWAS are conducted mainly in European subjects because data is way more available. I will also say that we ran the analysis in African population, and we also found the same exactly genomic region. So this specific effect can be identified across different ancestries. And that's it. This is all I have to say about erectile dysfunction. Thank you for listening.
Terrific. Thank you, Uri. Thank you to all of the presenters. Wonderful talks. Right now is the Q&A for the last 3 presenters. We are bumping up against time, so I'm just going to focus on the questions that came in from our audience. And one of those questions, I'm directing it to Uri. Uri, if there is a correlation with genetics of obesity, do you lose any of these ED associations if you control for condition on BMI? I guess BMI.
Yeah, so the true answer is I don't know. We conducted our analysis in all of us, and we meta-analyzed our results with previous publications. Therefore, we wanted to conduct our analysis as similar as possible to previous publications. These did not control for BMI. There are many factors that can be controlled for not only BMI, but prostate cancer or diabetes. And we did not control for any factor that was not controlled for in other studies. So we just controlled for very standard GWAS covariates, which are age, sex, not in this case, because this analysis was conducted only in men, and 10 genetic principal components. So I don't know the answer.
Okay, fantastic. Right. We are at time, a little bit over time, and we don't have any additional questions for the panelists, but I do want to thank you all for your presentations. I think they were very interesting and insightful, using the work of using the data, all of us data set. So thank you. Thank you all for joining us. You're our audience members. So right now is the keynote session. You'll see the presenter on the screen, Dr. Pradeep Natarajan. And actually following this session will be a short survey. So we would appreciate your feedback from that survey. So again, thank you for joining us in our poster session. Enjoy the rest of our All of Us Science Day.